Hi dear students, so in this video lecture we are going to study alkaline phosphatase test. This test basically is performed to check efficiency of pasteurization of milk. So let's start. Pasteurization is an essential process in production of milk which is safe and free from pathogen. In our previous lecture we have seen detailed aspects of pasteurization as well as sterilization. So pasteurization we perform for removal of pathogenic microorganisms from milk, and sterilization of milk we perform for complete removal of microorganisms and endospores or spores. So alkaline phosphatase is an enzyme which is naturally present in milk, but it get destroyed at temperature just near to pasteurization temperature. So alkaline phosphatase is an enzyme that is naturally present in milk, and this enzyme it get denatured the temperature just below the temperature we provide at pasteurization temperature. Okay. So alkaline phosphatase test is used to indicate whether the milk has been adequately pasteurized or whether it has been contaminated with raw milk after pasteurization. So what is aim of this test is that we can check efficiency of pasteurization, or if there has been any contamination of raw milk after pasteurization. So basically this test is performed, or this test is performed to check the efficiency after pasteurization. So this test is based on the principle that alkaline phosphatase enzyme in raw milk liberates phenol from disodium para-nitrophenyl phosphate and forms a yellow colored complex at alkaline pH. So what is the principle of this test is that the enzyme alkaline phosphatase it is previously or naturally present in raw milk. It liberates phenol from a compound that is disodium para-nitrophenyl phosphate, and it forms a yellow colored complex at alkaline means basic pH. So what you have to do, you have to measure intensity of yellow color produced. So intensity of yellow color produced is proportional to activity of enzyme. So the color intensity is measured by direct comparison with standard disk in Lovibond comparator. Okay. So what you have to do in this test, you have to observe yellow color. So yellow color is proportional to activity of alkaline phosphatases. So you have to compare yellow color with standard disk color through Lovibond comparator. So this test is not applicable to sour milk and milk preserved with chemical preservatives. Okay. So this test you can perform to check efficiency of pasteurization, and this test can be used only for pasteurized milk. So what are the apparatus required for this to carry out this test? So you have to need a water bath which is maintained at 37 degrees Celsius, and it should be thermostatically controlled. Then you need comparator. This is a special disc of standard color glasses calibrated in microgram p-nitrophenol per ml milk, and these are of size 2 into 25 millimeter cells. Then you need test tube of different test tube of size 16 into 1.50 millimeter with rubber stoppers to fit this test tubes, and you need 1 ml pipette, 5 ml pipette as well as 10 ml pipettes. Then you need filter paper that is of Whatman number 2 or equivalent, and you need litmus paper to check whether medium is acidic or basic. So overall these are apparatus and requirements that are needed to carry out this alkaline phosphatase test. So reagents. So how to prepare reagents for this particular test? So sodium carbonate and bicarbonate buffer. So this is a sodium carbonate and bicarbonate buffer system you have to prepare first. So to prepare this sodium carbonate and bicarbonate buffer system, you have to first dissolve 3.5 gram of anhydrous sodium carbonate and 1.5 gram of sodium bicarbonate in 1 liter of distilled water. So what you have to do, you have to take 1 liter of distilled water, and in that you have to dissolve 3.5 gram of anhydrous sodium carbonate. Then you have to dissolve 1.5 gram of sodium bicarbonate into that 1 liter water, distilled water. Then buffer substrate. So you have to dissolve 1.5 gram of sodium, 1.5 gram of disodium p-nitrophenyl phosphate in 1 liter of sodium bicarbonate buffer. So the buffer that you have prepared in first step, in that sodium bicarbonate buffer, what you have to do, you have to dissolve 1.5 gram of disodium p-nitrophenyl phosphate. Okay. So this solution is stable if stored in a refrigerator at 4 degrees Celsius or less for 1 month, but you can use this solution before 1 month, and you should store overall this solution in a refrigerator at 4 degrees Celsius, but a color control test should be carried out in freshly prepared solutions. Okay. Then what is procedure to carry out this alkaline phosphatase test? So in that you have to first pipette out 5 ml of buffer substrate into clean dry test tube, followed by 1 ml of milk that is to be tested. Then you have to stop the tube by stopper and mix inversion and place in water bath. So what you have to do, you have to take a test tube, then you have to add 5 ml of buffer substrate into that clean dry test tube. Then you have to add 1 ml of milk that has to be tested, and this test tube is covered by some stopper, and after inverting that test tube you have to place that test tube in a water bath which is maintained at 37 degrees Celsius. At the same time, place in a water bath a control tube. So you have to take a control and you have to keep this control with this our test tube, and in that how to prepare control, you have to add 5 ml of buffer substrate and 1 ml of boiled milk of same kind as the under test, that is pasteurized or homogenized, and which is of low fat content. So what you have to do, you have to check both test sample as well as control sample in this procedure. So after 2 hours, remove the tubes from water bath, invert each and read the color developed using the comparator and specialties. So what you have to do, you have to observe yellow color change, and you have to compare that yellow color of both test as well as control tubes with standard disc or special disc, and you have to observe color, or you have to match that color with standard disc. So the tube containing boiled milk control being placed on left of the stand, and the tube containing sample under test on the right. Record the reading which lie in between two standard color discs by adding plus or minus signs to the figure of nearest standard. Okay. So overall, what you have to do, you have to place control at left hand side, and test solution or test containing tube at the right side, and you have to score plus or minus by observing colors and by comparing these colors with special disc, and you have to calculate results, or you have to observe result and you have to put down result. So this is simple test that is performed to check efficiency of pasteurization. There is note, if artificial light is needed when taking these readings, an approved daylight is source of illumination must be used. So overall, what you have to do, you have to carry out all this process of alkaline phosphatase test in daylight. So this is overall background behind this alkaline phosphatase test. Once again I am repeating that this test is carried out to check efficiency of pasteurization, and you have to observe yellow color in this particular test. So thanks for this lecture. Okay.